Laura. Thank you very much for having me. It's very nice to be part of the first ever session of the Journal Club. So today we're going to discuss optogenetics and how it can be applied to chronophysiology and potentially even treat disease in the future. So what I'm going to do is very briefly introduce optogenetics, the fundamentals thereof. And I wanted to start by giving a very brief overview of action potentials. So what is an action potential? Most of you may already know what an action potential is. And it is the rapid rise and drop in voltage that occurs in cells that are able to generate action potentials, such as neurons, for example. But what happens during action potentials? So when cells are not stimulated, they are in resting potential, where the electrical charge outside the cell and inside the cell, the difference of the two, is around minus 70 millivolts. Then upon stimulation, which happens when neurotransmitters bind to receptors in the cell, they trigger depolarization, and this can lead to a threshold potential, which is the minimum difference in electrical charge, which can initiate an action potential. And then depolarization, mass depolarization keeps taking place by sodium ion channels opening, allowing an influx of sodium ions. Once the peak is reached, which is about 30 to 40 millivolts, these sodium ion channels start closing and potassium ion channels start opening, allowing potassium ions to leave the cell. This is what causes the drop in voltage, the repolarization, as shown in the diagram on the left. And it drops so much as to not only reach resting membrane potential again, but to overshoot it and reach a phase called hyperpolarization. Okay, so now what is optogenetics and what does it have to do with all of that? Optogenetics is a very recent method that has been developed at around 2004, if I'm not mistaken, and it uses light in order to stimulate genetically modified neurons by either exciting them or inhibiting them. These neurons contain protein ion channels that cause this, and one of those proteins is channel rhodopsin 2, which promotes excitation. So I wanted to, for a better understanding, I wanted to show you the pictures on the right. So on the top image, you can see petri dishes with green algae that contain channel rhodopsin 2. And you can see them how they move around the petri dishes in response to light. And then on the bottom picture, it's channel rhodopsins, and how when they're stimulated by blue light, they open, so they generate action potentials. And lastly, I just wanted to briefly go through how exactly optogenetics works. So how do we use it? And the way it does that is by obtaining the protein of interest from algae. Then the gene for this protein is put in a viral vector, which is then inserted in specific neurons in the brain. And these neurons start expressing this protein in their membranes. So then, once blue light hits those neurons that express this protein, they can generate action potentials. So this is very interesting because it can allow us to control certain behaviors, certain actions, such as movement, for example, which is why it could be useful in treating diseases in the future, like to control movement. And that's it for me. Thank you very much. Trying. Now, Kaveh is going to talk to you about chronophysiology.
Okay, thank you, Fiora. Can you hear me? Can you guys hear me? Okay. Okay. And now that we clearly understand the basis of optogenetics, I'll give you a summary of the basic principles of chronobiology. My objective with this presentation is that by the end of it, you will be able to understand what is a circadian rhythm, its molecular mechanism, which is very complex, but I'll try to summarize it in only 6 molecules. Also, we're going to review the organization of the circadian system and its 2 main components: the suprachiasmatic clock as the body's master clock, on the peripheral clock in the rest of the tissues and organs. And finally, I'm going to talk about a very important concept on circadian rhythms, which is circadian time. So let's get started. So, what is a circadian rhythm? Well, it is a ubiquitous feature of virtually all living organisms that consist of approximately 24 to 25 hours cyclic rhythm that persists in the absence of external entrainment, such as constant darkness exposure. And this concept was first documented in the 18th century by the French astronomer de Mairan. Yeah, 18th century. And he recorded his observation that the opening and closing of a heliotrope plant lives occur independently of the sunlight. But how did he discover that? Well, it was very simple. He took one of his plants, a sensitive, and created a constant darkness microenvironment, which means that he put the plant into his closet and observed that the leaves kept opening during the hours of sunlight and closed during the hours of darkness, independently of the exposure to light changes. When he finished with the experiment, the man concluded that the sensitive felt the sun without seeing it at all. And the cool thing here is that all of us have well established circadian mechanisms in almost every cell of our body. So we're like a plant, a sensitive but much more complex. Okay, that's not the best comparison, but you got my point. And how does the opening and closing of lips apply to humans? Well, we don't have lips, but we have many other physiological mechanisms. For example, glucose metabolism. And it's well established that the highest insulin sensitivity and glucose tolerance occurs in the morning, as well as many other hormonal changes and even blood pressure. And the best part of it is that we already know the molecular mechanism. So now let's talk about it. And the this is a summary because the complete molecular mechanisms includes many more molecules. But we have two main transcriptional factors. You can see BML1 and clock, and they need to effort dimerize to bind the evox sequences in the primary region of the genes that codify to pure and cry proteins. Once we have pure and cry proteins in the cytosol of the cell, they need to effort dimerase again, and in the particular case of PER, it has to be phosphorylated by a casein kinase so the dimer can translocate to the nucleus. And once the dimer translocates to the nucleus, it can bind to the same epoch sequences that clock and BML do to inhibit their own expression. So this is a transcriptional negative feedback loop. And we have two other transcriptional factors. The first is RIP-ERB, or also called NR1D1, and this is a negative transcriptional factor that will bind to the red response elements in the primary region of the gene that codifies to BML1. And this way it will inhibit expression of BML1. In the case of ROR alpha, it will bind to the red response elements in the primary region of BML1 and stimulate the expression of it. So we have three feedback loops in this mechanism. One negative of pure and cry that inhibits their own expression, another negative of RIP-ERB that inhibits the expression of BML1, and a positive of ROR alpha. Now, we divide the circadian system in humans in two main components: the master clock that resides in the suprachiasmatic nucleus of the hypothalamus, which is a group of around 10,000 to 20,000 neurons located at both sides of the third ventricle and above the optic chiasm. And the second component are all the peripheral clocks located in the rest of the body. A prevailing view of dissociation between the central and peripheral clocks is that of conductor and an orchestra. Each peripheral clock can play music independently, but the direction by the central clock helps the peripheral clocks play in harmony. And the suprachiasmatic nucleus receives direct retinal innervation via the retinal hypothalamic tract that connects the retina with the suprachiasmatic nucleus, and to ensure its synchronization to day-night cycles. And the suprachiasmatic nucleus clock projects to various brain centers, many of which contain local circadian clocks that direct behavioral. For example, feeding and fasting, and sleep and wakefulness, as well as autonomic and neuroendocrine circadian rhythms. And these systemic cues synchronize the local molecular clocks of peripheral tissues, kidney, heart, liver, etc. And now that the general organization of the circadian rhythm, we're going to talk about the master clock. And we can divide the suprachiasmatic nucleus into two components: a coronal subregion on the basis of retinal efferent connectivity and expression of various neuropeptides, with vasoactive intestinal peptide and gastrin-releasing peptide in the retinoreceptive core of the suprachiasmatic nucleus, and arginine vasopressin in the shell. And the retinal hypothalamic tract originates from intrinsically photoreceptive retinal ganglion cells that express a photopigment called melanopsin, which detects light. And once the retinal hypothalamic detects light, it would release a polypeptide called BACAP and glutamate that they both will bind to glutamate receptors types NMDA and AMPA. That will cause a depolarization of the core of the suprachiasmatic nucleus, and this will increase the spontaneous firing rate. And spontaneous firing rate are a bunch of action potentials of neurons. And once the stimulus increases the spontaneous firing rate, it will propagate to the shell of the suprachiasmatic nucleus, and this way activating the transcriptional translational feedback loop. And that's the molecular mechanism of circadian rhythm. Finally, we're gonna talk about this concept that is circadian time. Forget about these numbers because we don't have 12 to 12 dark light cycles or shifts. So what we have to understand is that when we're exposed to light, the expression of P and cry will be increased, as well as the ROR alpha. And at the end of the day, when the sun comes down and the concentration of these molecules is high in the suprachiasmatic nucleus, it will start to degrade to restart the cycle by the end of the day. Do you have any questions before we talk about the article? Oh, sorry. Okay. In the present study, the authors used optogenetics to selectively activate the vasopressinergic retinal ganglion cells axons in the suprachiasmatic nucleus, measured the influence of this on the electrical activity of the suprachiasmatic nucleus core neurons, and investigated the effects of a V1A receptor antagonist on evoked responses. So the methods, they used homozygous transgenic rat line expressing ABP enhanced reinforcement protein gene. And the rats were exposed to 12 to 12 dark light cycles. There were two groups, one that the exposure to light started at 7 a.m. and a shifted group of the light exposure started at 11 a.m. They used a recombinant adeno-associated virus vector carrying a conserved 1.9 kilobase ABP promoter that codified to channel rhodopsin 2 and the fluorescent protein M-cherry. For the intravitreal virus injection, they anesthetized the rats and used Midriatic in the eyes and injected 1 microliters in each eye of the recombinant adeno-associated virus vectors. For the study of the in vitro electrophysiology, they first anesthetized the rats and then they were decapitated. They took their brains and it was immersed in ice cold. Then it was sliced and put in different solutions. And for the view of the slices, they used an axioscope 2FS microscope. And then for the optogenetic stimulation, they used blue light. And finally, they applied the V1A antagonist to see if there was an effect in the evoked response. the analysis, they used 3 softwares: the Clanfit, Excel, and Prism. And the recording of the spontaneous firing rate before and after stimulation was recorded during 100, 200, and 500 seconds. The response assessment of the spontaneous firing rate, they measured the mean of the spontaneous firing rate at baseline and the spontaneous firing rate after the stimulation. And it was a responsive cell if the mean spontaneous firing rate increased, rated down 10%. For the statistical analysis, they used the Agostino and Person omnibus normality test for the deviations from normality. And to detect the changes in the spontaneous firing rate, they used the Ampere t-test. And here are the results. They only focused on the ventral lateral suprachiasmatic nucleus. Here we can see how the pipette injected the adenovirus. And shapes, the white shapes represent the neurons that responded to optogenetic stimulation, and the black shapes didn't respond to the optogenetic stimulation. And this is the immunohistochemistry of the cells that express the green fluorescent protein and the M-cherry fluorescent protein. So what they find? They found that the response of the ventrolateral suprachiasmatic to optogenetic stimulation really happened after 200 second stimulation. Here we can see how the spontaneous firing rate and the interspike interval increased after the blue light stimulation in the neurons, in responsive neurons. And this is an histogram of the interspike interval of the unresponsive cells in the optogenetic responsive cells. We can see a peak in the interspike interval, which means that the optogenetic stimulation actually increased the action potentials in the cells. After a 500 second stimulation, they detected a increase in the spontaneous firing rate as well. And here we can see the red shapes are the optogenetic stimulated neurons. And the blue shapes are normal neurons, and the orange shapes are non-responsive neurons. So here we can see how the spontaneous firing rate increased after the genetic stimulation. And the authors had to take the null hypothesis because they were looking for a response of the ventrolateral suprachiasmatic nucleus ABP expressing cells to genetic stimulation, and that didn't happen. After 300 second stimulation, there wasn't an increase in the spontaneous firing rate, and there was a decrease. And the use of antagonist of the arginine vasopressin peptide did work. They found that even when stimulating, if they use the antagonist, the spontaneous firing rate and the interspike interval decreased. So the conclusions of these articles are the following. The present study provides that evidence from communication between vasopressinergic retinal ganglion cells and ventrolateral suprachiasmatic neurons. So they are connected. The optogenetic stimulation increases the spontaneous firing rate of PB suprachiasmatic neurons, and the optogenetic activation of BP retinal ganglion auxins terminating the suprachiasmatic nucleus increase the electrical activity of approximately 30% of neurons in the ventrolateral suprachiasmatic nucleus. And because of the disruption of circadian rhythms in humans, it's associated with a whole variety of diseases and conditions, including obesity, cardiovascular disease, depression, and cognitive impairment. There is a great interest in understanding the mechanisms underlying circadian dysfunction to identify potential therapeutic targets. And that's it. So now it's time for questions or comments. Thanks, Alvaro. Okay. So any questions? I have to mention something about this. Even when the authors accept the null hypothesis, because vasopressinergic retinal ganglion cells didn't respond to optogenetic stimulation, the increase in the spontaneous firing rate activates the transcriptional translational feedback loop, so it causes changes in the peripheral clocks. So yeah. Theodora, I have one question for you.
Hi. Okay. I'll try my best to answer if I know the answer.
Is channel relapsing the only channel studied in optogenetic?
No, in fact, as I mentioned in my presentation, it's just that I mentioned it very briefly, I suppose. There are proteins that promote excitation and inhibition of neurons, and channel rhodopsin is one of those proteins that promote excitation. But there are also other proteins that promote inhibition of action potentials. I don't remember the names exactly, but there are more proteins than channel rhodopsin.
Okay, thank you.
Thank you.
So please ask your questions. Is there any other question? Okay. Doris, you said you had a question. Okay. Well, if there's no more questions, thank you for joining today. Thank you, Theodora, for your presentation. And I guess that's it for today.